All right, welcome to The Explainer. Today, we are jumping into one of the biggest biological challenges, of the last 50 years. We're talking about cracking a code that literally determines how life itself is built. And guess what? It's a puzzle that AI has finally solved, and yeah, it's changing everything.
So you've got a spider's web, an antibody that can save your life, and the stuff in your stomach that digests your lunch. What could they possibly have in common? Well, the answer is simple, but at the same time, incredibly complex. It all boils down to some amazing microscopic machinery.
Yep, proteins. They are the absolute workhorses of life. They do just about everything inside our cells. And their function, their superpower, comes down to one thing: their intricate, three-dimensional shape. You get that shape even a little bit wrong, and the whole machine just breaks.
Okay, so this reliance on shape, it creates a huge problem, one that has stumped scientists for decades. See, the weird thing is, the entire blueprint for that super complex 3D shape is somehow hidden inside a simple, one-dimensional string of its building blocks, the amino acids. And that puzzle is what we call the protein folding problem.
To put that in perspective, imagine you have a string of letters a mile long. That's it, just the letters in order. Now your job is to predict the exact ridiculously complex origami sculpture that string will fold itself into. That's the scale of the challenge we're talking about here.
So, how did we used to do this? Well, for a very long time, figuring out these shapes was, let's just say, a slow, painstaking grind. Scientists had some brilliant methods, for sure, but they were just so inefficient, especially when you think about the millions of proteins out there in nature.
So you had your experimental methods, right? Things like X-ray crystallography. They were the gold standard, but man, were they slow. We're talking months, sometimes even years for a single protein. So naturally, we turned to computers to speed things up. But the early models, they really struggled. They were okay if they could find a known protein to copy from, like a template. But if it was something new, their accuracy just plummeted. It created this massive bottleneck in biology.
So to break through this bottleneck, the world of AI came up with two totally different, really powerful ideas. It was kind of like a classic clash of philosophies. One approach was inspired by evolution, nature's own creativity, and the other, it was inspired by the way our own brains learn.
All right, let's look at the first one, evolutionary computation. The best way to get your head around this is to literally think survival of the fittest, but for protein shapes inside a computer. The AI just generates a whole bunch of possible shapes and then lets them compete and mutate over generations. The good ones survive, the bad ones don't. Simple as that. So how does that work practically? First, you have to represent the protein as data. Then you need a scoring system, basically a judge that uses physics to say how good or stable a shape is. And then comes the fun part. The AI uses what they call crossover and mutation, mixing and matching the best bits of the successful shapes to hunt for even better ones. And it does this over and over for thousands of generations. Okay, now for the other camp. Their philosophy was completely different. Instead of evolving a solution from nothing, they thought, why not just learn from all the examples we already have? And this, of course, is the world of neural networks. The idea was to feed an AI the massive database of all the protein structures scientists had already solved and let it learn the rules of folding for itself. Now, in the early days, these networks didn't actually build the 3D structure, not directly anyway. They were more like a guide. What they did was predict something called a distance map. You can think of it like a chart that says, hey, this part of the protein chain should be close to that part. And that map gave scientists the key clues they needed to piece the final structure together. So for years, you had these two camps, evolutionary computation and neural networks, and they were both making steady progress. But it was slow, incremental gains, and then everything changed. It happened at this competition called CASP. You can basically think of it as the Olympics of protein folding. So in 2018, this team from DeepMind shows up with something called AlphaFold, and it just blows everyone away. The accuracy was way beyond anything anyone had seen before. But that was nothing. 2 years later, they came back with its successor, AlphaFold 2, and it delivered a result that didn't just cause a stir, it completely shocked the entire scientific community.
Just look at these numbers. Seriously. A score over 90 here is considered as good as what you can get from a real-world, multi-year lab experiment. AlphaFold 2 didn't just beat the old computer methods, it soared past them with a median score of 92.4. For the very first time, an AI could predict a protein shape as accurately as the painstaking work that took scientists years. That 50-year-old grand challenge, it was basically solved.
So, what was the secret sauce? How did they do it?
Well, AlphaFold 2 had this radical new design. It wasn't just predicting that 2D map anymore. It was what's called an end-to-end system. That means it was actually building and tweaking the final 3D structure right there, inside the network, thinking about the whole puzzle in 3D space from start to finish.
And this breakthrough, it was so massive that in 2024, its creators got the call from Stockholm. They were awarded the Nobel Prize in Chemistry for this work. And that Nobel, it wasn't just recognizing a cool achievement. It was signaling the dawn of a completely new era in biology, an era powered by AI.
Okay, so the problem is solved. What now? What does this actually mean for the future of, science and medicine? What new doors did this just blow wide open?
Well, think about it like this. It's like we've suddenly been handed the blueprint for almost every single tiny machine in the entire living world. This just puts everything into overdrive. Drug discovery, designing brand new treatments, even engineering totally novel proteins from scratch. We're talking about custom-built microscopic machines that could, say, eat plastic in the ocean or hunt down cancer cells with laser precision.
But, and there's always a but, right? This incredible power brings a whole new set of challenges. For one, these neural networks are often black boxes. We know they get the right answer, but we don't always know why or how. They also take a ridiculous amount of computing power to run, which is a huge barrier for a lot of researchers. And then, of course, you get into the ethics of it all. Being able to design new proteins is amazing, but it also raises some serious questions we need to think about.
And this actually brings us full circle, back to those two AI philosophies we started with. Because there's a really interesting trade-off here. On one hand, you have the neural networks like AlphaFold. The accuracy is just world-changing. But on the other hand, you have those older evolutionary methods. Sure, they're less accurate, but their process is way more transparent. You can actually understand how they got to their answer. They're explainable.
And you can bet the innovation is not stopping here, not by a long shot. The next big thing people are talking about are protein language models. Now, you know how AI like ChatGPT learned language by basically reading the whole internet? Well, these models are doing the same thing, but for biology. They're analyzing billions of protein sequences to learn the fundamental language of life. It's like they're learning the very grammar of biology itself.
So yeah, this whole revolution that was kicked off by solving the protein folding problem, it's really just the beginning. It feels like we've finally opened the book of life, and the biggest question now isn't just about what's already written inside, but what we're going to write in the next chapter. Thanks for joining this explainer.